Welcome to Aquatic Biology. Imagine you are standing by a beautiful, clear lake. Beneath that calm surface is a complex, invisible world of dissolved gases and solids constantly interacting to sustain life. Today, we are going to explore these hidden chemical drivers. We will learn how fish breathe, how lakes protect themselves from acid rain, and what happens when a lake gets too many nutrients. Let us dive in. To begin our journey, let us talk about the breath of the lake, which is dissolved oxygen. Why does this matter? Just like us, fish and other aquatic organisms need oxygen to survive, but they rely entirely on oxygen gas that has dissolved directly into the water. This is not the oxygen bound to hydrogen in the water molecule, but independent oxygen molecules mixed in. The amount of oxygen the water can hold changes based on physical factors like temperature and pressure. If these levels drop too low, the lake experiences hypoxia, which can lead to devastating fish kills. It is the most critical parameter in aquatic biology. Now that we understand what dissolved oxygen is, let us look at how we can calculate its concentration using Henry's law. Conceptually, this formula tells us that the amount of gas dissolved in water is directly proportional to how hard the atmosphere is pushing that gas into the water. In the equation, C represents the concentration of the dissolved gas. The symbol Kh stands for Henry's law constant, which changes depending on the water temperature and the specific gas. Finally, P is the partial pressure of the gas in the atmosphere. If the atmospheric pressure P increases, the concentration C will also increase, assuming the temperature stays constant. Before we move on, let us see this formula in action with a concrete example. We want to find the theoretical dissolved oxygen concentration in a cold mountain lake. We are given the atmospheric partial pressure of oxygen as 0.21 atmospheres, and Henry's constant as 1.3 times 10 to the negative third moles per liter per atmosphere. We choose Henry's law because it directly connects atmospheric pressure to water solubility. By multiplying our given constant by the partial pressure, we calculate the final concentration. The result is 2.73 times 10 to the negative fourth moles per liter. This number tells us exactly how much oxygen is physically available for the fish to breathe under these specific conditions. Having calculated the oxygen at the surface, let us visualize how oxygen behaves deeper down. This diagram shows a stratified lake dissolved oxygen profile. On the vertical axis, we have lake depth in meters going downwards. On the horizontal axis, we have the dissolved oxygen concentration. Notice the top layer, the epilimnion, has high and steady oxygen levels because it's in contact with the air and sunlight. But as we cross the dashed line into the middle layer, the metalimnion or thermocline, the oxygen concentration drops sharply. By the time we reach the deep, dark bottom layer, the hypolimnion, the oxygen is nearly depleted. This visualizes how physical stratification creates distinct living zones for aquatic life. Moving from oxygen, we must now consider another vital gas, carbon dioxide. Why should we care about carbon dioxide in a lake? While oxygen is for breathing, carbon dioxide acts as a master chemical regulator. When it dissolves, it reacts with water to form a weak acid, which then breaks apart into bicarbonate and carbonate ions. This creates a reversible chemical chain known as the carbonate buffer system. This invisible chemical dance is crucial because it acts as a shield. Without it, a simple rainstorm could cause wild, lethal swings in the lake's pH. It also provides the essential building blocks for aquatic plants to photosynthesize and for creatures like snails to build their shells. To quantify this protective shield we just discussed, we use the total alkalinity formula. Conceptually, this formula calculates the total capacity of the water to neutralize acids by summing up all the basic components and subtracting the acidic ones. In this equation, ALK stands for total alkalinity. The terms in the square brackets represent molar concentrations. We have the concentration of bicarbonate ions and the concentration of carbonate ions. Notice the multiplier of 2 in front of the carbonate ion. This is because each carbonate ion can neutralize 2 hydrogen ions, making it twice as effective. The last 2 terms represent hydroxide and hydrogen ions. If the concentration of bicarbonate increases, the overall alkalinity increases, making the lake much more resistant to sudden pH changes. Let us apply this buffering formula to a real laboratory sample. We are given a bicarbonate concentration of 2.0 times 10 to the negative third moles per liter and a carbonate concentration of 0.5 times 10 to the negative third. We are asked to find the approximate total alkalinity. Because hydroxide and hydrogen ions are negligible here, we simplify our formula to focus only on the dominant carbonate and bicarbonate terms. We substitute our values, remembering to multiply the carbonate concentration by 2. Adding these together gives a final answer of 3.0 times 10 to the negative third equivalents per liter. This relatively high value physically means the lake has a strong, healthy buffer system, well protected against acidic pollution. Now that we have covered gases and buffering, let us shift our focus to the dissolved solids, which act as the vitamins of the lake. Why are these important? Just like a garden needs fertilizer, microscopic algae at the base of the aquatic food web need dissolved nutrients to grow. The most critical of these are macronutrients, phosphates, which contain phosphorus, and nitrates, which contain nitrogen. These dictate the biological productivity of the entire lake. However, balance is key. If human activities like farming dump too many of these dissolved solids into the water, it triggers a destructive process called eutrophication. This causes massive, toxic algae blooms that eventually die, rot, and consume all the dissolved oxygen we discussed earlier. To understand how these nutrients control growth, we use the nitrogen to phosphorus ratio formula. Conceptually, algae need both nitrogen and phosphorus to grow, typically in a specific biological proportion known as the Redfield ratio, which is about 16 to 1. This simple equation calculates the relative proportion of these 2 critical nutrients in the water. R is the ratio itself. The numerator, N in brackets, represents the total concentration of dissolved available nitrogen. The denominator, P in brackets, represents the total concentration of dissolved available phosphorus. If the value of nitrogen increases significantly while phosphorus stays the same, the ratio R increases. This physical change indicates that phosphorus has become the limiting factor, holding back further algae growth. Let us test this ratio with a water sample suspected of agricultural pollution. We are given a dissolved nitrogen concentration of 320 micrograms per liter and a phosphorus concentration of 10 micrograms per liter. We need to calculate the ratio and determine the limiting nutrient. We use our ratio formula because it directly compares the availability of these 2 elements. By dividing 320 by 10, we get a final ratio of 32. We then compare this to the ideal biological Redfield ratio of 16. Because 32 is much greater than 16, there is an overwhelming abundance of nitrogen. Physically, this means phosphorus is the limiting nutrient. adding even a little more phosphorus will trigger a massive algae bloom. We have covered a lot of ground today, so let us review our key takeaways. First, we learned that dissolved oxygen is the fundamental breath of the lake, essential for survival, and highly dependent on physical conditions. Second, we saw how carbon dioxide creates the carbonate buffer system, acting as a chemical thermostat to protect the lake from lethal pH swings. Finally, we discovered that dissolved solids, specifically nitrogen and phosphorus, are the biological engines of the ecosystem, but must remain balanced to prevent destructive eutrophication. You now have a solid understanding of the invisible chemistry that dictates aquatic life. Great job!